Next, I'd like to introduce Sarah Anzick, PhD. She is a senior research specialist in the Research Technologies branch at Rocky Mountain Laboratories in Hamilton, Montana, where she utilizes genomic-based technologies to study allergy and infectious diseases. Dr. Anzick holds an MS from University of New Mexico and a PhD in molecular oncology from George Washington University. Dr. Anzick assisted in the Human Genome Project at Los Alamos National Laboratories, and then she moved to the National Human Genome Research Institute in 1994, where her research focused on cancer genomics. She has been with the National Institutes for Health for over 20 years. Most recently, using her personal time and resources, she participated in the ancient DNA sequencing of one of America's first indigenous people, a 12,600-year-old Clovis boy found in western Montana. Dr. Anzick.
Well, I'm deeply honored to be here today, and I'd like to thank the organizers for including me in this important event. Over the years, I've faced numerous challenges, tremendous burden, and personal conflict on a subject that has traditionally sparked debate and contention among tribal people. It's a story of human remains, and I've been invited to share with you my personal journey and experiences on an ancient DNA study, which was done to help us understand the genetic legacy of one of the earliest inhabitants of America, a 12,600-year-old Clovis child from western Montana. The story begins in 1968 when two construction workers obtained permission from my father to extract sandstone from an outcrop on my parents' property in Wilson, Montana. The landfill was to be used for a local school construction project. Using a front-end loader, the workers uncovered one of America's most significant archaeological finds, but at the time they didn't realize the importance of what they had accidentally unearthed. The finders recovered over 100 stone and non-human osseous tools that were completely covered in red ochre, an iron hematite substance commonly used in ceremonies. Within this assemblage, they also found the partial and fragmentary remains of a subadult human, estimated to be approximately 2 years old. The artifacts were recognized as belonging to the prehistoric Clovis culture, which I'll describe in the next couple of slides. Carbon dating of the materials determined that the site was approximately 12,700 years before present. This is the end of the last glacial maximum when Ice Age animals were still in existence. In 1979, some of the artifacts were featured on the cover of the National Geographic. The Clovis culture is the name given to prehistoric hunter-gatherer culture whose spear points were first identified near Clovis, New Mexico in 1933. The Clovis people are believed to be the descendants of the first people to enter the Americas around 15,000 years ago. Shown here, the Clovis culture appears to have originated south of the ice sheets that covered Canada, as no Clovis artifacts have been found in Siberia or Alaska. It's apparently unique to the North American continent. A different complex of archaeological sites have been found south of Panama. Listed here are 4 of at least 6 South American sites, which radiocarbon date to Clovis, but they lack the diagnostic Clovis tools and technology. From 13 Clovis sites in the United States that have been radiocarbon dated with precision, the data suggests that Clovis is a relatively short-lived period, approximately 400 years. Shown here on the x-axis is the calibrated ages of the Anzick human remains and the osseous tools relative to other Clovis sites. The human remains radiocarbon dates are estimated approximately 12,600 calendar years before present. This is near the end of the Clovis period, and the osseous tools calibrated ages average 12,850 calendar years, or toward the beginning of the Clovis era. The reason for this age discrepancy is unknown, but it's been postulated that the osseous tools represent heirlooms that have been passed down through the generations. Oops. The Anzick site is exceptional in that it is the only known Clovis site which contained human remains. Now I'd like to share with you my personal journey of privileges, coincidences, and challenges that I have faced over the years. I was not quite 2 years old when the site was accidentally discovered on my parents' property, approximately the same age as a child. In my undergraduate years, I had aspirations of going to medical school or following in my father's footsteps and going to vet school. But I did a summer internship at Los Alamos National Labs and discovered my love for research. I was privileged to be able to participate in the Human Genome Project in 1992, work of which led to my first publication. In 1994, I was recruited to the National Human Genome Research Institute, where my research focused on cancer genetics and genomics. I had a very early exposure to the human genome and what we could learn from this amazing book of instructions. In 1999, the human remains were returned to my family. They were in the hands of the original archaeologist. And this is when I recognized that I was uniquely positioned as a scientist and the daughter of a landowner to potentially glimpse into the ancient past by understanding and trying to gain an understanding of the genome of one of America's earliest inhabitants. Because the remains were found on private property, NAGPRA didn't apply. But to glean insight into the perspectives and the feelings of Native American communities over genetic studies, I visited 2 tribes who were in closest proximity to the site. Some tribal members were supportive and others were not. It became very clear that it would be difficult to obtain a consensus among tribes. I had no clear guidance of where to go, but I felt an obligation to humanity. These remains I felt were a gift and had a story to tell. Because they were 13,000 years old, finding the lineal descendants of these ancient Clovis individuals seemed impossible without some genetic analyses, particularly for an individual from a culture whose archaeological record vanished alongside the mass extinction of Ice Age animals. Furthermore, some theories posited that Clovis people originated in France or Spain, an out-of-Europe hypothesis. Dr. Mike Waters from Texas A&M approached me with an interest in pursuing the ancient DNA work on the Anzick remains through collaboration with Professor Eske Willersov's lab at the University of Copenhagen in Denmark. Eske's lab had made remarkable progress in the ancient DNA technology. His lab had successfully sequenced the first ancient genome, that of a 4,000-year-old Paleo-Eskimo from Greenland, an amazing technological achievement. When the genome of this 4,000-year-old Sakak individual was compared to contemporary populations, the results showed that this individual is extinct in the Americas. His closest relatives are the Arctic peoples of the Siberian Far East. And so I embarked on the project, using my personal time and independent of my position at NIH. Importantly, my involvement in the project meant that I could ensure that the remains were handled carefully and with respect, using the minimal amount of material possible. So I traveled to Denmark to perform the first extraction and look at the mitochondrial DNA haplogrouping. The initial genetic mitochondrial DNA screening of the Clovis individual showed that Anzick belonged to the root of haplogroup D4H3A. Interestingly, this haplogroup matched the mitochondrial haplogroup of 2 other ancient skeletons, On Your Knees Cave from Southeast Alaska and Ancient 939 from British Columbia, suggesting it was more frequent in ancient times. The D4H3A haplogroup is also found in a Han Chinese individual. Interestingly, it has not been found in any ancient or living individuals on the northwest coast after approximately 6,000 years before present. Throughout the Americas, it is extremely rare and only found in about 1.5% of living native populations, most of which are in South America. Mitochondrial DNA, however, only gives us the matrilineal descendancy. And so to get a more comprehensive and informative perspective of the population, the nuclear genome was sequenced. This was many, many years of technological developments to make this happen, but the Anzick genome was finally sequenced, and we learned that the Clovis child was male. And using a statistical method for estimating relatedness, and to place Anzick in the context of present-day human populations, the Clovis genome was compared to 143 worldwide modern-day populations, including 52 Native American populations. As shown here in red, which is a measure of relatedness, with red being the most closest related, Anzick 1 showed highest relatedness to all 52 Native American groups than to any other Eurasian population. Interestingly, Anzick was more closely related to 44 Native populations from Central and South America than to 7 Northern Native Americans from Canada and the Arctic. Thus, Anzick is most closely related to an estimated 80% of Native American tribes from which DNA exists for comparison. It's important to note that in the United States, it is still a white spot on the map as no DNA is available for comparison. The genetic data are consistent with a simple tree-like model from a common ancestor. There is an early divergence between Northern Native Americans and Southern Native Americans that appears to go back to pre-Clovis times. Anzick sits on the Southern Native American branch. Outgroup F3 statistics was also used to compare the Anzick genome with 3 previously sequenced ancient individuals: the 4,000-year-old Sakak individual from Greenland, a 40,000-year-old human from Tianwan, China, and a 24,000-year-old individual from Malta in Siberia. Anzick is more closely related to modern Native American populations than to the Sakak individual. When Anzick was compared to the 24,000-year-old Malta individual, Anzick is closer related to Eastern Asians and Native Americans, but shares about 1/3 of his genome with the Malta individual. Malta is closer related to Western Eurasians. Likewise, Anzick is more closely related to Native Americans and East Asian populations than to the 40,000-year-old Tianwan individual. These data are consistent with a dual ancestry model, in which Native Americans descended from 2 ancestral populations, East Asians and Western Eurasians. These results also support a pre-Clovis occupation of the Americas and refute an out-of-Europe hypothesis. Last fall, as Shane mentioned, Professor Eska Willislev and Shane Doyle traveled to various tribes in Montana to inform them of the results of the ancient DNA work. While none of the tribes objected to the work, most of the tribes requested reburial of the ancient remains. The Anzick family has agreed, as this is our moral obligation, to put this child to rest. He's given us a generous gift. A private reburial ceremony is scheduled for June 28th. In my final slide, I illustrate that ancestry is not all we can learn from ancient DNA studies. Ancient DNA work can reveal important historical events, including the emergence of infectious diseases, as observed in the 5,300-year-old ice mummy Otzi, who is shown to have the oldest case of Lyme disease, which is caused by ticks infected with the Borrelia bacteria. We can also gain insight to the evolution and history of modern diseases, including heart disease, diabetes, allergy, and cancer, for example. There are many, many people I wish to recognize for these efforts, including efforts that have gone into the upcoming reburial plans. But most importantly, I'd like to thank my family and my parents and my husband for their unending support throughout this process. I'd also like to thank Dr. Shane Doyle. He's been instrumental in serving as a liaison for this work. I've had to take a lot of time away from my job and my family, and I'm very grateful to have this opportunity to be here today and to present this. Thank you. Oops.